Hello everyone, my name is Samchita. I'm from Odisha. I'm currently doing my bachelor's in NIT Raulkela in Department of Life Science. So again, hello everyone and welcome to today's session. And we have with us Professor T.S. Suryanarayanan, who is currently serving as professor and director of Vivekananda Institute of Tropical Mycology, which is a unit of Ramakrishna Mission Vidyapeeth, Chennai, India. Professor Suryanarayanan has experience in teaching mycology, plant physiology, plant ecology, and plant pathology. He also conducted a workshop in endophyte mycology for graduate students at Arizona State University. And coming to his distinctions as honors, he has 140 publications and 13 book chapters. He has 5,505 citations, and his H index score is 40. According to the worldwide data prepared by Stanford University, he is named in the list of top 2% of scientists in mycology from India. He is also the consultant at Center for Next Generation Technologies in Adaptive Agriculture in ICAR and World Bank funded microbiome-enabled seed priming project, which is from University of Agricultural Sciences, Bangalore, India. He was also awarded Fulbright Nehru Senior Scholarship Fellowship to conduct research for 6 months on study the feasibility of using endophytic fungi for biofuel generation. This was a collaborative research with Professor Venkat Gopalan, Department of Biochemistry. He was also awarded Fulbright Fellowship for studying fungi associated with cacti of Sonoran District at Desert at Arizona State University. He is also an honorary research associate at Fredericton and St. John's campuses, which is at University of New Brunswick, Canada. He is also a research supervisor at University of Münster, Germany. He is also visiting scientist at Department of Molecular Pathology, George August University, Germany. He is also a deputy visiting research fellow at Rajiv Gandhi University, Itanagar. He has many research collaborations with Germany, Estonia, USA, Finland, IIT Madras, ISC Bangalore, a lot more. So without any further delay, so I request Dr. Suryanarayanan to enlighten us with his wisdom. So please.
Oh, thank you, Samchita. So I just want to make sure all of you are able to hear me.
Yes, sir.
Excellent. So what I will do is I straight away share the screen and yeah, are you able to see this first slide?
Yes, sir.
Okay, so let me begin my talk. So so before I proceed further, thanks again for that nice introduction. So I'll straight away go to the talk. As I mentioned earlier, the first half of the talk will be with reference to the, what are antibiotics and why should such antibiotic molecules be produced by microorganisms? And they don't produce such metabolites so that we humans can use it for curing our disease. It never happens in nature. In nature, as you all know, an organism spends energy and produces molecules only for its survival. Okay, so what are the functions of antibiotics, and what are the different types of antibiotics, and what happens in nature, and how this how bacteria develop resist, or before that, how antibiotics kill other microorganisms. What are the mechanisms by which antibiotics work? And then how bacteria defend themselves against antibiotics produced by other bacteria, and how this has become a global health problem. So this is what we're going to see. And finally, towards the end, some possible solutions to tackle this or to manage this global health crisis. This is what we are going to look into. So you can feel free to stop me during the talk if you have any questions, and I try to answer them if I'm able to. So here, let's go to the next slide. Okay, so here it is. What is an antibiotic? It's a chemical, it's a molecule produced by one microorganism. When we say microorganism, generally I'm including bacteria and microfungi, okay? Acinoacids and others as well. So it's a chemical produced by one microorganism to kill or to stop the growth of another microorganism. Now, you take an example of as a habitat, an environment, like for example, soil. Now, in a soil, many different microorganisms survive. So there will be competition among these different microorganisms which are present in the soil. There will be competition for space and also for the food that is present in the soil, the nutrients that is present in the soil. So let's say there's a lot of competition and fight. So how do these organisms, microorganisms in the soil, avoid this competition? How do they reduce the competition among themselves? Now, some microbes grow faster than the others, so they quickly grow. As soon as some nutrients are present in the soil, they grow very quickly. Then they reproduce very fast, and they produce spores or whatever, which will be blown off, and then they when the spores fall on a suitable place again, they germinate. So they just have a very short life cycle. They are called R-selected species. So they're very fast, and that way they escape competition. The others, how they escape competition is they eat, or let us say they use nutrients which are not eaten by or which are not used by other co-occurring microbes. So you can see in a soil sample, if there are different species of bacteria, some of them would depend on the, would be using the nutrients which are not used by others, and some of them will use a nutrient and then they excrete whatever, let's say the byproducts, which may become the food for some other microbes. So there is a lot of this interaction. So they can survive the competition by not depending on the same type of nutrients. And the third one is by the production of antibiotics. So some of these microorganisms, they produce molecules which will stop the growth or kill the competing species which occur in the soil. So managing competition or reducing the competition by producing antibiotics, that process is called antibiosis. Okay. Now, so antibiotics are produced in nature by microorganisms as a mechanism to avoid competition. And it was found, as you know, penicillin was the first antibiotic to have been used by humans. So some of these molecules which are produced by these microorganisms for avoiding competition can also kill the bacteria that cause disease in humans and that cause disease in animals and also in plants. Therefore, we use these antibiotics for curing our diseases. So that's how antibiotics come into medicine. Right. Okay, so let's go to the next slide. Now, I told you about, we are taking soil as an example, as an environment. The same thing happens in different environments, including, let's say, water body, or on the surface of plant, near the roots, or in the dung of an animal, anywhere where there are microorganisms, including our gut, where there are millions of bacteria that are present. So every place where there are microorganisms present, there is going to be competition, and there is going to be different types of reactions and interactions between them to avoid competition. But we will take soil as an example. So here, let us see in nature what happens. How does antibiotic help the producer of antibiotic to survive competition? You take here. Can you see the arrow of my mouse?
Yes, we can see it.
Okay, good. So you take this place here. This is a soil sample in which we are seeing different species of bacteria, microorganisms. Let's say B is one species, A, X, and Y. And then there could be many more, but let us limit ourselves to these 4 different types of species. Now, you assume that there is another bacterium, which is, let us say, species YAM, which is different from all this. And, let's say that it shares the same nutrient that is with B. So it eats the same food in the soil which is eaten by B. But A eats a different nutrient, Y utilizes a different nutrient, and X utilizes a different nutrient. One second.
So in the meantime, people, can you all hear me?
Yes, Sri.
So in the meantime, let me please introduce here in the audience Professor Gopala Vinjamuri, who...
Yeah, sorry for the break. Okay, so can you hear me? Yeah.
Yes, sir. Yeah.
Okay, so now see, so M and B share the same nutrients. Let us say M enters, but because M and B share the same food, and if M enters, B can attack it, not by producing any antibiotic, but actually it can grow on it and kill it. It's a parasite. So there is no chance, as long as B is there in this soil sample, there is little chance of M coming in and establishing itself. The moment it comes in, B can detect it, and it can go and parasitize that and kill it. So M is removed, and now B has no competition in the soil. It can live happily because A, X, Y, and B, they don't share. There is no competition between them for nutrients because they take different types of nutrients. Okay. Now let us proceed here. This example. Now what happens here is, in the same setup where these bacteria are there, another species of bacterium, let us call it K, it comes in. How does it come in? Well, you can imagine that sometimes spores of bacteria land in the soil through, they're carried by wind or bird droppings or whatever. It just comes in, and this K, it has a gene which produces an antibiotic, and that antibiotic kills Y. Okay, which is competing with K for the same food. So if Y is not there, K can come in, and it won't, even though it has a gene, it will usually not produce an antibiotic because Y is not there. But if Y is already there, or if Y comes in, then K produces an antibiotic to kill it. Now, that way, K is superior to Y because it is having a gene to produce antibiotic, and it can win the competition with Y. Good. Now, it so happens, it can also happen that this antibiotic can also kill B. Although it doesn't share the nutrient with it, probably the molecule has a wider range of action, so it can remove B. If B is removed, then M can come in because there is no B here, and B cannot parasitize this, so M can come in. So here is an example how production of one antibiotic can change the microbiome or whatever, the constitution of microbial species in a small sample. Are you able to understand this? Can I go to the next slide?
Yes, sir.
Yes, sir.
Yes, sir.
Questions? No. Okay. Yeah. Now, as you all know, penicillin was the first therapeutic antibiotic, the antibiotic that was used in medicine during the around that period, World War II. Let's see how it works. Now, the cell wall of bacterium is made up of molecules called peptidoglycan. And these peptidoglycan molecules, you can imagine these molecules as something like bricks placed one above the other. And then they are attached to each other by bonds, chemical bonds. Now, what happens is when penicillin is present, the penicillin molecule will enter the bacteria, and then what it does is it breaks the, it prevents the chemical bondage between the peptidoglycan molecules. It's like removing the cement between the bricks. So the cell wall becomes weak. And when the cell wall becomes weak, water from the surrounding environment, maybe from blood or in the intestine, whatever, water will rush in. And the cell wall, so because of the entry of too much water, the internal osmotic pressure of the cell will increase. But because the wall is weakened by penicillin, the wall will break in different places, and then the protoplasm of the bacterium would come out, and our immune system will take care of it, and then it kills the bacteria. So this is how penicillin works. It doesn't directly kill the bacterium, but weakens the cell wall, and that results in the death of the bacteria. Well, that is how penicillin works, but different antibiotics work in different ways. For example, so you have different classes of antibiotics acting in different ways to kill. Let's see here, you take, there are some antibiotics which will inhibit the ribosome function, and thus protein synthesis in the bacteria would be affected. That means several enzymes may not be produced, and that will lead to the death of the bacterium. Tetracycline and chloramphenicol are these examples for protein inhibiting antibiotics, protein synthesis inhibiting antibiotics. And then polymyxin is the one antibiotic which will enter, and then it will, just like penicillin breaks the cell wall, this one will disrupt the membrane, the plasma membrane, lipoprotein membrane. So the membrane structure is damaged, and that will lead to the death of the bacteria. Penicillin and cephalosporin, we have already seen the mechanism of action. And then let's see, there are some antibiotics like rifamycin and fluoroquinolones, which will enter, and then they inhibit the replication of DNA, the circular DNA, the major circular DNA. So the bacterial, the nucleic acid synthesis would be inhibited, and that leads to the death of the bacteria. Now, these are all, all this polymyxin, chloramphenicol, tetracycline, penicillin, etc. All these things are all antibiotics produced by different bacteria, but there are also synthetic antibiotics like sulfonamides. These are not synthesized by any bacteria, but they are synthesized by chemists in the laboratory, and that can also function as antibiotics sometimes. Here is one example, sulfonamides, which will inhibit the formation of certain very important compounds within the cell. Here, this one will inhibit formation of synthesis of folic acid, and then that can damage the cell, and that leads to the death of the bacteria. So you see how different classes of antibiotics work in different ways to effectively destroy bacteria. Now, we have already seen that antibiosis is the production of antibiotic by a bacterium to stop the growth or to kill another bacteria. Now, that is like a warfare. And whenever there is a warfare, the enemy, I mean, this is the, let's say this is the bacterium which produces the antibiotic. And this is the bacterium that gets affected. It's not going to sit simply and allow itself to disappear or die. During the course of evolution, the bacteria which are susceptible to the antibiotic will also evolve mechanism to manage or to defend themselves from the antibiotics. How does this happen? So let's see the next one. How bacteria resist antibiotics. So it's really like a chess game. One bacteria moves one coin by producing an antibiotic. The other bacterium which is susceptible to the antibiotic, which can get killed by the antibiotic, will move another coin to defend itself. So this warfare will go on. It is going on and on and on for millions of years, evolution. So then what happens? There will be a lot of one particular bacterium produces an antibiotic to kill another bacterium, and then in that population somehow some bacteria develop methods to manage or to fight the antibiotic, and then the antibiotic produced by the original bacterium is useless. Then that antibiotic producing bacterium will slowly evolve to produce a better antibiotic. Then the killing will take place. So this will go on. That's what is expected in nature. So let's see how bacteria resist antibiotics. One method is the bacterium which is susceptible to an antibiotic can develop protein pumps which will be located in the plasma membrane, and these pumps are called efflux pumps. What they do is, as soon as the antibiotic molecule enters, it will be selectively pushed out by this pump. So there is no accumulation of the antibiotic within the cell. Only if the antibiotic molecules accumulate, they can do their work. They can go and damage something and kill the bacterium. But this pump will selectively throw it out. It won't throw other things. It will selectively throw only the molecule, the antibiotic molecule. And it's a one-way pump. It will not work this way. It will only work. So it's called an efflux pump. And this is one method by which the a bacterium can survive when antibiotic enters. The second one is the bacteria can develop thick wall so that it decreases the number of antibiotic molecules that enter the cell. And with the decrease in the number of antibiotic molecules, they may not be effective. We need a particular concentration of a chemical to work, isn't it? So if you are having a fever, you take at least 500 milligram or 650 milligram of paracetamol. What if you take the same paracetamol but 1 milligram? It's not going to work. So that's what happens here. So there is a decrease in the antibiotic molecules that enter the cell. Then that is the second method. The third method is, let's say the antibiotic comes in and then it has a target. Like, for example, I told you somewhere here. Yeah, let's say this sulfonamide comes in. And what is it? How does it kill the cell? It will inhibit the production of compound like folic acid. So when folic acid, this will prevent the production of folic acid, and then the bacterium dies. Now, what the bacterium can do is, let sulfonamide come in, instead of producing 100 molecules of this sulfonamide, let us say it produces 10 million molecules of sulfonamide. I'm sorry, of folic acid. So the sulfonamides can break as many as they want, but there is going to be a plenty of folic acid, so the effect is minimized. That is another way by which the target molecule is overproduced. You get it? So the bacteria is not affected at all. You may have 500 molecules of this antibiotic, each one attacking one molecule, and 500 molecules of the target molecules are affected. But what if the bacterium produces 5 million molecules? No use. So that the antibiotic will be useless. Or it can also, modify. This is the target molecule. The antibiotic attaches itself to this target molecule and then prevents the function or breaks down this molecule, so the bacterium dies. But what the bacterium will do is it will slightly modify it, maybe attach a methyl group or OH group. I'm just guessing. It can slightly modify it chemically so that the antibiotic cannot recognize that molecule. So you have a modified target, and even if the molecule antibiotic come in, they have no method of working. And finally, another method is the bacterium can produce special enzymes which will break down the antibiotic molecule. That's all. So here is an enzyme which will inactivate the antibiotic molecule. Okay. Well, so also, even if a bacterium is defenseless against a particular antibiotic, when that defenseless, susceptible bacterium is exposed too often to a particular antibiotic molecule, or when it is exposed to too much of that antibiotic molecule, it is possible that that susceptible bacterium will slowly develop one of these resistant mechanisms and would become resistant to the antibiotic. That is in nature. If there are no questions, I can go to the next slide. Are there any questions?
Any questions? Friends, if you have any questions, please raise your hand. Okay, so that way I will know that we have questions. You have questions. Okay,
that's a good idea. Yeah. Okay. So I'll proceed. Okay. Now the next thing that we are going to see is how the susceptible bacterium, the mechanisms of escaping antibiotic we have seen, but how do they become resistant? Suddenly, how do they become resistant? That's what we're going to see. There are some mechanisms. One is by mutation. You know that bacteria divide very fast. Maybe it depends on the species, but if you take E. coli, maybe 21 in one division in 20 minutes, and they rapidly increase their population. So, in general, when bacteria divide, they divide, and then they produce a number of cells. So whenever the bacterium divides, the DNA of the bacterium also divides. That divides first, and then this bacteria divide. So whenever there is DNA division, there is going to be some mistakes in the DNA sequence. So it's not it tries to have exact replica of the DNA in every young cell that is formed. But during this, there are sometimes, even though there are correcting measures, as you know, their errors can happen. And usually, in about 10 hours, the bacteria would have a very huge population, and within that 10 hours, there could be more than 300 DNA mutations, 300 DNA mistakes. Many of these mutations may not be advantageous, and so many of the mutant bacteria usually die. They cannot survive. But by chance, out of these 300 mutations in 10 hours, one mutation can provide a mechanism for one bacterial cell to become resistant to an antibiotic. If not in that 10 hours, In the next 10 hours, you are going to, in the next 10 hours, the entire population is going to double. You know that logarithmic growth is there in bacterial cell division. So a huge number you get within a couple of days. There are going to be many mutations, and it is possible that one of the mutations gives the bacterium one of these mechanisms. And then, so that bacterial cell will become resistant to an antibiotic. So here, let us imagine you have a number of bacteria, particular species of bacteria, and here is an antibiotic. A is a very effective molecule in killing this particular bacterium. A will be there, A will enter, it will kill almost all the cells, but during the cell division, let us say that one of the bacterial cells has a mutation because of which this A antibiotic is not able to kill this. And so in the next few generations, all these original susceptible bacteria would die, but this one will multiply, and you get a huge population, because when this multiplies, that same mutant DNA will be transferred to the daughter, and then from there to the daughter, from there to the daughter. So within a few hours, you see a buildup of a population of bacteria, which are all resistant to that antibiotic. They all come from this particular single bacterium. The population of the susceptible bacteria will be reduced because the antibiotic will kill them, and the population of the resistant resistant strain would increase.
Sorry to interrupt you, sir. Are you able to take a question now, sir, please?
Yeah, of course.
Sure. Okay. So Avantika, you had a question, it looks like.
Dr. Narayanan, we are very glad to have you here. I have a question from the first slide. Like, I have zero idea about bacteria, and also this might be a very naive one, but yeah, here goes. Like, how does M and B know that they are competitors, that they feed on the same nutrient, or how does A, X, Y know that B isn't a threat to them? Like, are they capable of deciding who is the enemy and who is not a competitor? Like, are they capable of doing that?
Yeah, good question. Okay, so yeah, we are made to think like that. I mean, we are used to thinking like that. Actually, there is no such predetermined thinking. So what happens is, let's take you take this soil. Okay, so here is a soil sample in which all these bacteria are there. They don't, the Y doesn't know that. That's one way of looking at it. It doesn't know that it is not competing. It has its own food, it survives, it goes ahead. This has its own nutrient, it goes. This has its own nutrient, this has its own nutrient. Okay, so they are independent of each other. The moment M enters, M doesn't know that B is there, B doesn't know M is there, but M comes in, and M will start, eating the nutrient that is also shared by B. And when M and B are close together, because the nutrient source is coming down, M is using the nutrient, so the amount of nutrient is going to come down, and that will function as a signal for B to induce the production of an antibiotic. And then the antibiotic, when it is produced naturally, when it is produced, it will be in high concentration near the cell, and it will diffuse. Simple physical formula, from high concentration to, it will diffuse, and when it diffuses, if M is around, M would get killed, and when M population goes down, there is more food, B can multiply more. And it is not always the entrant gets killed. It is also possible that the entrant can come in, the entrant can produce a better antibiotic than B, and M can kill B, eliminate B, and can stay there.
Understood, sir. So when B feels stressed due to not getting enough nutrients, then it starts producing the antibiotic.
Yeah, that's a signal. Yeah, that's a signal. Yes. So many of these organisms have genes for producing different types of secondary metabolites, including antibiotics, but they don't produce it when it is not necessary, because production of a metabolite requires energy. They don't waste energy. So there should be some sort of a triggering needed to activate the gene so that they produce. If you're working with bacteria, if I take a bacterium and put it in a culture medium, you can try. It will usually not produce the number of, some secondary metabolites will be produced, some, but many of them may not be produced. The genes will be silent. Why? Because it's a pampered situation. You provide that bacterium with lot of food, and there is no other bacterium there in the culture. It's a pure culture. So there is no need to compete. It's happy, plenty of food, no fight needed, no neighbor to come and take the food away, and then a lot of air, and so everything is fine. You are providing it at the right temperature, but it's happy. It doesn't produce unnecessarily secondary metabolites. The moment you bring in another bacterium there, then you see inhibition taking place. One will inhibit the other, which we do not know which one will, but by looking at it, you can see inhibition. Then there is a signal which will induce one of the bacterium to produce the antibiotic. Got it?
Yes, sir. Thank you so much.
Oh, welcome. Shall we proceed?
Not really, sir. We have another question, sir, if you don't mind.
Yeah, please.
Soham, do you want to go real quick, please?
Yeah. Namaste, Suryanarayanan, sir.
Yeah.
I wanted to ask that how bacteria senses this kind of stressors. Like first thing you told us that lack of decrease in the nutrients makes it trigger its resistance or the antibiotic
Production. Yeah.
production.
Hmm.
So how does it senses that yeah, we have this much stock of nutrients, and now that whole stock is reducing? How would it know?
No, see, first thing, reduction in the concentration of nutrients is just one of the signals. And then how does it know? Okay. When there is enough nutrients, the bacterial species will grow and then divide, and then each cell will be original. You can roughly, each new cell will be the size of half the size of the original cell, and then each one will grow to a new, to the original size, and then so this will go on and on and on, and within a few hours, number of bacterial cells of that particular bacteria, the population builds up. When the population builds up, it is not even necessary for another bacterium to come and compete the food. Even this own bacterium, because of multiplying, there is going to be reduction in the food. At that time, the bacterium would take rest. It will produce, if there are bacteria which can form spores, so they will form spores and they will take rest. Spores will not eat food. The metabolism is very low. They're just resting, like seeds of plant. And then that's because there is no food. And when the spore gets dispersed to some other place where the food is there, again they will start growing. So that's how it works. But when another competing species is there, the situation is different. And then there are a number of triggers. One is reduction in the food. Not only that, the presence of the other bacteria. There are going to be interaction between the two. When the two bacteria are together, some of the metabolites which are leaked out by the new bacterium can be detected by this original bacterium, and then it can produce an antibiotic. Okay, so there are, you can look at the net. There are what triggers the production of antibiotics? You can see that there are methods by which this can happen. Remember that good question you asked. This interaction I'm talking to you is between, I always say bacteria, plural. Bacterium is singular, as you know, but it can be also between a bacterium and a fungus. It can also be between a fungus and a fungus in the soil, in our intestine, on the surface of a leaf, on the surface of a fruit. Can happen anywhere where there are a population of microorganisms this type of interactions can take place. Okay, and I gave you reduction in food as just one of the signals. There are many other signals which can induce antibiotic. The bacteria can sense that and produce antibiotics. Okay, perhaps there is a new one species comes in, it it eats the food and then it produces some secondary metabolites which are leaked out. One of those could be a signal for the another species of bacterium to detect, oh, there's another species here, let me synthesize antibiotic. So that's only one of. Can I proceed now, or do you have any questions?
We'll just have one more. We'll take one more question, sir. If you
Sure,
don't mind.
sure. No problem.
Rudra, do you want to go real quick? So before you ask your question, Rudra, say what your name is, what you do, and where you're from, because that will give a perspective to, Yeah, go ahead, Rudra, please.
Good evening, Suryanarayanan, sir. I am Rudra Narayan Sahu. I'm a first year postgraduate student in botany from Jamia Hamdard, New Delhi. I'm based in Puri, Odisha. I have this question: can we consider antibiotics to be synonymous with secondary metabolites?
Sorry, I can't hear.
Can we consider antibiotics to be synonymous with secondary metabolites?
Yes, they are secondary metabolites. Yeah, they are.
And second question is chlorophyll a primary metabolite or secondary metabolite? Is chlorophyll a primary metabolite or secondary metabolite?
His question is chlorophyll? We can't hear you properly, Rudra. You need to fix it. First of all, right. And the question, I guess, is, correct me if I'm wrong, Rudra, is chlorophyll a primary or a secondary metabolite?
Yes, sir.
Chlorophyll.
Yes, sir.
Oh, wow. Chlorophyll. Yeah. Okay. So how do you distinguish primary and secondary metabolites? Primary metabolites are basically needed for all the metabolism, survival, and all that. Secondary metabolites are, one of the old definitions is secondary metabolites are not necessarily needed for the survival of the organism, but they are needed sometimes for, like this competition, for increasing the fitness of an organism. For some of these things, secondary metabolites are needed. So chlorophyll is definitely a primary one, without which photosynthesis cannot take place.
Okay.
Okay, thank you. Yeah, so we could probably go ahead, sir, please. Thank you.
Okay, excellent. So where was I? Yeah, okay. So if a bacterium is, even if a bacterium is defenseless against an antibiotic, it can become resistant to that antibiotic when it is exposed to too much of that antibiotic or too often to that antibiotic. Okay. We know that antibiotics are given to us patients when we have some infection. So there is a dosage. Every antibiotic has a particular dosage strength, 250 milligrams or 500 milligrams, and the doctor said twice per day or 3 times a day for 4 days, for 5 days, whatever. So we have to follow the strength as well as the time. And then next time when I get the similar fever, I feel that, oh, I know this, I don't have to go to the doctor. I know last time I took this antibiotic and I became all right, so let me take this antibiotic again. When you do that, we are exposing our flora, intestinal flora. Our stomach and intestine has millions of bacteria. So we are exposing our bacteria to this particular antibiotic too many times, and many of the bacteria there would die because of this antibiotic. But this can happen, this one I told you, somewhere here, somewhere, this can happen. Like it can develop a mutant, and that mutant can fill your intestine, and next time when you take that antibiotic, it's useless. So this can happen anywhere, wherever there is a group of microorganisms. Okay, now so so one of the mechanisms by which can you hear me? Because there is a power cut here, but don't worry, I'm on a backup Can you hear me?
Yes, sir. Yeah.
Okay.
Yes,
So I told you one of the methods by which bacteria can develop resistance to an antibiotic is by mutations. And the mutant accidentally, certain mutants are not killed, but and then they increase the population, and that's how the resistant strain of a particular bacterial species develops. So that is by mutation. Now we are going to see, is there any other method? Yes, there is another method, very common and probably very effective, by transfer of resistant genes. You know that bacteria have a circular DNA molecule. In addition to that, small circular DNA molecules called plasmids. A bacterium can have a few plasmids, which are DNA molecules, double-stranded DNA molecules, which are not the main DNA molecule. There is one main DNA, and a few plasmids may be there, are usually present. The plasmids also have genes like, just like the main DNA, and they have a few genes. Plasmids have a few genes. And the antibiotic resistant genes are usually present in these plasmids. First thing. And second thing is, a plasmid can replicate it independent of this DNA. This main DNA replication takes place when the bacterial cell divides, but the plasmid can multiply irrespective of that. These are basics. Now let us go. So there are whenever, TB, there are TB patients, so who can be treated with an antibiotic called isoniazid. So you treat them with isoniazid for a few months, then their TB is cured, because the TB bacterium that they have, that they are infected with, is killed by isoniazid. There are some other TB patients where the isoniazid treatment is useless. Isoniazid doesn't kill the TB bacterium that their body is having. They are infected with Mycobacterium tuberculosis, which causes TB. They are infected with the bacterium which is resistant to isoniazid antibiotic. So they give rifampicin. So TB patients, the moment you see a TB patient, you have to test and see which of the two antibiotics is effective for that person. Whether he's infected with isoniazid resistant bacterium, then you give him rifampicin. If he is infected with rifampicin resistant antibiotic, then you give her isoniazid. Okay. Now, here is a TB bacterium which is killed by isoniazid, but it is not killed by rifampicin. The reason is it has a plasmid. In that plasmid, there is a gene, and that gene is the one that is responsible for giving the resistance to rifampicin to this particular TB bacterium. Okay. Now, here is another TB bacterium. It has a plasmid. That plasmid has a gene here, which maybe produces an enzyme which will break down isoniazid. So, if you give isoniazid to this patient, it doesn't cure her, but you can give rifampicin, it will cure her. Okay. So, we have two pathostrains, two disease-causing strains of the same TB bacteria. One that can be killed by isoniazid, not by rifampicin. The other one that is killed by rifampicin antibiotic, but not by isoniazid. Okay. Now, everything is fine. What could happen is, let's have these two big TB patients together in a hospital next to each other in a bed. And then let's say they cough, the phlegm in the air, the bacteria mix, or the catheter that they use for both the patients. So the catheter is used for removing the urine because the patient is lying down. So, or the syringe used for when they come together up. the bacteria, some bacteria sticking to the tube or to the injection syringe can come together. And when they come together, this can happen. So here is the plasmid, which carries the resistance for rifampicin, this bacterium. It can multiply and it can go. These two bacteria can form a channel between this, and then this plasmid carrying this gene can go here. Now this bacterium becomes resistant to both the antibiotics. So a new superbug is created, which is now if I get infected with this TB bacterium, you can give me rifampicin, I cannot be cured. You can give me isoniazid, I cannot be cured. You can give a mixture of these two, no use. Okay, so this is how multi-drug resistant bacteria evolve. MDR, multi-drug resistant bacteria in nature. This is how they evolve. Now the next thing is the title says of my talk is antibiotic resistance, a global health crisis. It's not my own title. You can see this is now a very common title. WHO uses this. How can it be a global crisis? That's a huge, you know, it's really frightening that global. Okay, we have seen whether we use antibiotic or not, whatever. Evolution of antibiotic resistance among bacteria and fungi is natural. It will go on, right? In anywhere, when the interaction we have already seen, it's like a chess game. The bacteria can slowly mutate, but it is happening. Resistance to antibiotic is happening in nature. We can't stop it. But what we humans have done is we have speeded up tremendously this process of evolution of antibiotic resistance. That is why it has become a crisis. I'll give you one example. You take this bacterium called Staphylococcus aureus. It's a common bacterium. It causes skin infection whenever they have a cut, you know, while shaving or when we fall and when we cut the skin, it can cause a skin infection. It can also cause respiratory infection. It was easy to treat. Well, now due to this type of mutations, because we have been using that antibiotic too often, even for just a cut in our skin, we've been using that, and then it is also by this transfer of resistant gene and all that. Now this Staphylococcus aureus has become resistant to antibiotics. Not one antibiotic. It has become resistant to methicillin, amoxicillin, penicillin, oxacillin, and cephalosporins. So it is a multi-drug resistant bacteria. It is difficult to cure. What was a very simple disease, if somebody had a Staphylococcus aureus respiratory infection of the upper respiratory tract or the lung, you could treat it with an antibiotic, and within 4 or 5 days the person should be all right. But now you can give any of these drugs, no, that is resistant, and a simple respiratory infection, it cannot be cured. By the year 2050, according to World Health Organization, multi-drug resistant microorganisms would cause 10 million deaths annually, and that will result in a global cost of $100 trillion US. So it's a major threat. Okay, now I said we humans were responsible in speeding up this antibiotic resistance, which is taking place in nature, but at a slow pace. We have speeded it up. And that's how this global crisis, I told you that. How did we speed it up? One, overuse. Taking two antibiotics too often, the two for wrong reason, So we are treating patients with a lot of antibiotics, and then again and again, and for wrong reasons and all that. So we are here. Antibiotic pressure we give. We throw a lot of antibiotic to a population of bacteria, and because of this mutation can take place, and then anything could happen, and the bacterium could develop one of these methods to, or even two or three methods to become resistant. So that is one. Then contamination. So we are not using antibiotic only for curing human diseases. We also use antibiotics in agriculture for protecting plants against bacterial infection. Rice plant also. We use antibiotics, and that antibiotic would go to the soil, and in the soil there are bacteria. This antibiotic would, from the plant, it will go to the soil, and the presence of that antibiotic can cause a lot of changes in the microbiome there, and it could lead to development of resistant bacteria. And then abuse. In many places, you can just go to a shop and without any doctor's prescription, you can get an antibiotic. Just say, I want this rifampicin, they'll give you. It's not so in other countries, at least in the West. They go to other extreme. You can't even see a doctor. Huh? That's also pretty not a good way to manage. But anyway, so abuse, prescription is not needed. Self-medication, as I told you, I get a disease, I get some sort of a problem, I go to a doctor, he or she gives me an antibiotic, and I become all right. And next year I develop similar thing. I don't go to the doctor. I do it myself. I know. I think I know. So say last time it cured me, so I'll go and buy my own medicine and eat it. I mean, take them. Then mismanagement of hospital waste. Well, this is really bacteria already resistant to different antibiotics which are present in blood, urine, catheter, waste, exchange resistant genes between unrelated bacteria when they are brought together. The management of hospital waste is a very critical thing. If they are not properly managed, there are different types of waste, as you know. Bandages and syringes, and then operation instruments, catheters, and which are actually inserted into the patient. Then the tubes, respiratory tubes, which are introduced into the patients, all those things. So, and if all this waste, then the blood soaked or phlegm samples, etc., after operation, they will be having these bacteria. And if they are, let's say, just an example, we are operating on a patient, and then the patient has bacteria resistant to some antibiotic A, and then the blood-soaked cotton is not burnt but thrown away, and then the same thing happens from another patient who is having bacteria resistant to antibiotic B, and then these are all dumped in a place. Somebody collects them. There are contractors who collect this waste, and they are supposed to burn them. If they don't do that, they dump it in a place. This happens, and then the two cotton blood-soaked cottons come together. Then there's going to be exchange of genes and evolution of superbugs. Unrelated, the gene which is resistant gene which is present in TB bacterium, as an example, could move by the plasmid transfer to not another TB bacterium. It can easily do it to another TB bacterium, but it can go to a cholera bacterium. Totally unrelated, taxonomically unrelated bacterium. So this is called horizontal gene transfer. Taxonomically unrelated bacteria can acquire genes from each other by this horizontal gene transfer, and that is how superbugs develop. What are superbugs? Okay, there are types of superbugs. Generally, they are classified as MDR, which is multi-drug resistant bacteria. What are they? These bacteria are resistant to at least one chemical of 3 antibiotic types. We have seen some are here. Different types of antibiotics out there, right? There are a few more I didn't mention. There are different types. This is chemically different from this, and this is chemically different from this. If a bacterium is resistant to this and this and this, then you call it multi-drug resistant. There are extensively XDR, extensively drug-resistant bacteria. So these are even more dangerous. That is, they are resistant to at least one antibiotic of almost all the chemicals. So it would be resistant to this, this, and this, and this, and this by horizontal gene transfer and mutation and all. Then you have PDR, pan drug resistant. They are resistant to all known antibiotics, most dangerous. And from MDR, XDR can develop. From XDR, MDR, PDR can develop. From MDR, PDR can develop. So the World Health Organization has listed the most dangerous MDR pathogens. These are all those bacteria which are very difficult to control, and these are high risk. They are developing already in nature. They are already becoming MDR, and they are going to be very difficult to control. These are all very common bacteria. Helicobacter pylori is pretty common in the stomach of everyone, but if they become multi-drug resistant, it could be really a problem. And now the situation, okay, your question would be, so what is it? Okay, if the bacteria become resistant to penicillin, this, why don't we discover new antibiotics, right? That way we can manage the drug-resistant ones. But no new antibiotics are available. In the last 20 years, no new antibiotics have been. Why? Pharmaceutical companies, they don't want to invest in novel antibiotic research. Reason? They have to spend at least 15 years and several billions of dollars to get one effective molecule. You take the time taken, see the time taken and the money spent just to get one effective new antibiotic. Okay, if they get that, are they assured of getting back the money? Will they make a lot of gain? No, because even if new antibiotics are produced, government regulations will not permit the use of new antibiotics widely. Whenever a new antibiotic is produced, the government would ask the doctors, don't use it. Because if you use it, quickly the bacteria would become resistant to even that. So you have to delay the evolution of resistance. Therefore, do not prescribe it unless it's really necessary. So very few people would be prescribing it, very few patients would be taking it. And so if my company spends so much, what is the money I get back? And again, even if these antibiotics are, we never take antibiotics throughout our life, right? We take antibiotic till the disease is cured, maybe for a week, maximum for 10 days. But if it is a BP blood pressure drug or anti-cholesterol drug, we take it throughout our life. So if you are a drug company, drug manufacturer, you would prefer to manufacture this, because if you find one patient, he's going to or she's going to take it throughout her life. If you produce an antibiotic, the patient will use it only for a week. So, and they try to screen instead of trying to get antibiotics from nature. The scientists started looking at synthetic chemical libraries, whether from that we can make novel antibiotics, but they have not been successful, so they are giving it up. And then so the so-called novel antibiotics which are in use today are mere chemical modifications. You take penicillin, many bacteria have become resistant to penicillin. So what the chemists or the pharmaceutical chemists do is they slightly modify that molecule. make it, penicillin G or something, try to change it a little bit. And then it will work for some time, but quickly the bacteria would develop resistant to that because it's easy. Already they're resistant to penicillin, and this is only a slight modification of the original penicillin. So it becomes damn easy for the bacteria to become, so they quickly develop resistance to these so-called novel antibiotics, which are only merely chemical modifications of those molecules to which already resistance have been developed. Okay, so are there any hands up questions? Shall we proceed?
Yes, please. I mean, it looks like they're all quite.
Okay.
Yeah,
okay. So now comes this question: how do we solve this? Actually, solve is not the right word. How do we manage it? One is what WHO recommends is improve prescribing practices. The doctors have been told that the prescription practices should be more proper, and then we try to synthesize more synthetic antibiotics, although so far they have not been very successful. And then because the multinational pharmaceutical companies are not willing to invest money in this, the funding, it is suggested that funding should come from public and from governments and non-profit organizations, and the research should go on to find out more new antibiotics. And then there are other methods you can refer to. I'm just adding this. I'm not going to explain this because nanotherapy is something which I don't know much about. Of course, phage therapy, antibodies, use of bacteriocins, and deep learning approaches. This is another thing I'm not very, I don't know much about anything about this. So there are, I think, these are methods that have been suggested to look for novel antibiotics. But hunting for novel chemicals from microorganisms of hitherto unstudied environmental niches, that seems to be an interesting method. So you look for natural antibiotics from microorganisms of habitats or environments which have not been studied in detail so far. So this point, let us elaborate this point. And see, you take for example, I'll show our work. So, just like we have bacteria within our body in the intestine, we call them the endobiome bacteria. Just like that, plants also have bacteria and fungi within their roots and within their leaves. They don't cause disease, so you don't see any disease symptoms. They're absolutely normal looking leaf. You just pluck any leaf which looks pretty normal, and then you can isolate fungi and bacteria from inside. So that is called the endobiome of plant. So there are fungi inside the plant. We call them endophytes. Endo means within, phyte plants, within plants. So many people have started working on these fungi, and in one of our works, we had this brown seaweed, which marine algae, which grows in the sea. We collected that, and also from mangrove leaves. We found a fungus here. This is a fungus called Fusarium, which we have got from this particular brown alga. This is magnified about 500 times under microscope. This produces a chemical which kills malarial parasite, Plasmodium falciparum. Oh, this is wrong. It's just Plasmodium falciparum. This is repeated again. Which is that Plasmodium falciparum, which is actually resistant to chloroquine. Chloroquine is the drug which is used for curing malaria and but there are malarial parasites which are resistant to chloroquine. But we showed that the endophyte from here could kill even such resistant bacteria. Kindly excuse me for just
Absolutely, yeah. I mean, I would rather like to introduce somebody now, but I think I'll probably swallow it and wait for a little longer, because it'll be really hard for me to handle if there's another interruption. But that's okay. We'll wait. Yeah, we'll wait. So we, yeah.
I'm really spooked about how many antibiotics I took the last time I was infected with COVID. And the last time I'd gone to the dentist for my dental treatment, I had tons of antibiotics.
Yeah, with COVID, it's not antibiotics that you take. It's more of I don't know, vitamins and minerals, because they say zinc helps quite a bit, magnesium helps quite a bit. Is what they're saying. It's not. It's more than the antibiotics. It's probably you take. You take the vitamins. Yeah. So okay. So I'm going to introduce somebody, sir. If this is well. Yeah,
I'm back. Yeah. Sorry, I was not here.
No problem. Sir, is it are you able to take a couple of questions, sir, please? No? Sure. Okay. So Shrishti, if you want to go first, please, and please introduce yourself, what you do, so that gives perspective to Professor Suryanarayanan. Yeah, please go ahead.
Good evening, sir. Thank you once again for this webinar today. My name is Shrishti Parikh. I'm currently living in Bangalore, and I'm doing my second year bachelor's degree in Mount Carmel College. So my question was from a couple of slides ago about how humans have been accelerating resistance to antibiotics. So with the concept of self-medication, most of the time when patients are affected, they purposely, don't choose to go to the doctor, maybe due to financial situations, or they just don't want to. So this leads to them self-medicating. So what can we do in this scenario, right? Because we can't, force them to go to the doctor, and it can become a problem. So how do we battle this situation?
Very good question. So, there is a feeling, even among educated, affordable people, that antibiotics are something like magic. They are magic, maybe for the first time, but it's always. So how do we educate? What do I do? So I tell people who are, let's say, the boy who supplies milk to my house, the boy who supplies newspaper, the lady help who comes to my house. I always tell them, never use the medicine that has been given by a doctor again unless you meet the doctor again. Well, we can say that, but whether it will be respected, I'm not sure. So people need to be educated. Yes, let's say, okay, one way is to write what do you call this? General articles in newspapers and all these Twitters and Facebook stuffs and things there. The doctors themselves can write without using technical terms and all that. They can educate the public and say that it is not a good way. They don't have to talk about all this evolution of multi-drug resistant and all that, but they can impress. They can impress people. They can impress the patients. A few doctors do that, but generally not all of them do it. So I have a feeling that I may be, we are entering a very a thin ice here, thin ice. So doctors should be, practicing doctors should be given, and surgeons should be given a something like what teachers, college teachers have, some sort of a refresher course on how to deal with patients. Don't deal with them as just machine. They come here, I have a complaint, okay, give this, get out. No. So they must have a sort of a more human relationship, which will also inform. A time has come when we cannot treat doctors as demigods as we used to do, as our parents used to do. Oh, like in like some doctor, you go to him, he has got, what do they call in Tamil? They say Rasiyana doctor. If you go to doctor, so those things never help now. So because science has advanced so much, and the doctor should also be given, I don't know whether they have a separate, they should have a separate course on how to deal with this sort of dangers and how to deal with patients. As you rightly said, all patients need not know about the technicalities of these things, but they should be warned. Maybe general articles by some young people like you can do that.
Thank you, sir. Good question. Saloni, do you want to go real quick, please?
Yes, thank you. So my name is Saloni and I am first year master's student. I'm from Delhi. My question is, our grandmothers have been telling us to take certain type of, things when we are ill, and they have antibiotic nature. So my question was, generations have been consuming it and we didn't develop resistance towards them. Or, I mean, like even like common cold, if we drink the turmeric milk or something, like whatever, we have not developed a resistance. Why it is so?
See, one way, the way I look at it is, the population, correct me if I'm wrong, the population of bacteria that existed, grandmother, that existed 50, 60 years back are different from the bacteria that exist today, right? Because of all these processes. So there are some people whom I know, they say, I never take antibiotics. They think that, yeah, I may not, I may say that I'm going to be, no, I won't take any antibiotic. I will depend on, like you said, the traditional medicine or some other method, acupressure, acupuncture, whatever. I'm not, okay. So I don't take antibiotic. Good. But I go to a shop to buy some chips or banana. Guy next to me, he has been taking antibiotic, he has been treated, he has a lot of multiple resistant bacteria in his system. He sneezes, he coughs, and I breathe, I get him. If I don't take, it's not going to save me. So the time has come when we have to depend on such chemicals. Because of the evolution of bacteria, because of the evolution of microbes, there in 50, 60 years back, the bacteria did not have such a pressure. Even if you go to the net, even the discoverer of penicillin, who is it? Who's that?
Alexander Fleming.
Exactly. Oh, I got confused with Ian Fleming. Okay, Alexander Fleming, he himself says that this antibiotic will be useless after 50 years. He predicts that. So the type of microorganisms that are causing disease in us, the same disease, the TB that is caused today, is different from the TB that was caused 50, 60 years back. If you had analyzed, you take a genetic analysis of those TB bacteria 60 years back, and now you're going to see a lot of difference. There are going to be plasmids with more mutant genes, more antibiotic resistance in the current month. That's why it is becoming difficult to cure. It's not easy. And then there are other things, like for example, let's say 50 years back, if somebody's kidney failed or somebody's liver failed, there's no alternative. The person has to die. Today we have organ transplant. When we have organ transplant, when an organ is transplanted, what happens? The body tries to reject it because it's a foreign. So the doctor advises a drug to reduce the immunity. The immunity reduction will reduce the body's nature to discard the foreign organ. But at the same time, the common immunity is reduced. So I'm going to get infected by all sorts of bacteria. Earlier, I was not susceptible to this. Therefore, the doctor gives antibiotics. There are many people with the organ transplant survivors. They take a lot of antibiotics. In their system, there is going to be mutation. In their system, there is going to be development of multi-drug resistant, more chances of multi-drug resistant bacteria developing in their system. Then from their body, it can escape. And it can come, sorry, it can infect a normal person. So a time has come when we cannot live without dependence on antibiotic. And the time has also come when almost all the antibiotics currently in use are useless. It's going to be really tough. That's why we call it a crisis.
Thank you, sir. Thank you, Saloni. So, Janvi, do you want to go real quick, please?
Yeah. Hello, sir. I'm Janvi Deshmukh. I'm currently in my last years of my bachelor's and my subject is zoology. I had a question, and correct me if I'm wrong. Why are AMPs not able to replace antibiotics? Because I think even AMPs are naturally available abundantly, and I think they work similarly by destroying the cell membrane of the pathogen.
Which one you're talking about? I mean, which chemical or whatever? Which method?
Antimicrobial peptides.
Peptides. Yeah, and there are antimicrobial peptides which also work as antibiotics. Yeah, to which resistance can develop. Yeah. There are other things. Okay, I'm coming to that now. I'm in this slide. I'm going to talk about that. Okay, so can I proceed?
Yes, sir. Please.
So look at this. This is from our lab. So this is, it gives me some strength that I'm not just talking some general talk, picking up things from Google. Here we see. So this one. So here, this is the about malarial parasite and its life cycle. These are the red blood corpuscles, and mature red blood corpuscles do not have nucleus, so that means they don't have DNA. But if it is infected with the malarial parasite will divide. You know about the life cycle there, and the malarial parasite will contain the DNA. So if I take a blood sample of a patient and I use a particular dye, a fluorescent dye, which will attach only to DNA, and then it will fluoresce. So suppose I take a blood sample from a normal person and then inject this dye, I won't see any fluorescence because all my red blood corpuscles or mature red blood corpuscles do not have a nucleus and there is no DNA. But if the person is infected with malaria, some of the red blood cells will be having this Plasmodium parasite, which has DNA nucleus, and it's going to fluoresce. More fluorescence, more bacteria are, more malarial parasite are there in your blood, right? That is the assay that they use. It's called Cyber Green assay. Here you see that Cyber Green is the dye that they use. Cyber Green is the dye that they use for it. So here, when we grow this, when we took this fungus from this brown seaweed and then grew it in culture and got the metabolites produced by that, and then when you put it here, it brought down the fluorescence, which means it was killing the malarial parasite, and it did not damage the RBC. And the malarial parasite which we used or there is a strain which is resistant to chloroquine. Chloroquine is a common drug which is given for malaria, treating malaria. And here is a strain which is resistant, but our chemical could reduce it and it doesn't kill the RBC. That is very important. I may get a wonderful chemical from a microorganism, a new antibiotic. There are many things to come, that's why people find it very difficult. There are many people who have found new antibiotics. It works in the lab, but you cannot give it to a patient because it not only kills the bacterium, it also kills the human cells. It's useless. Or it should be, it is, the dosage. You have to give it in a small amount so that it will work. But what is the point? Suppose I found an antibiotic which is excellent, but I have to take 1 kilogram of it per day. It's ridiculous. It can damage other things. So dose response, and then ability not to damage normal cells, and then then the chemical can go in. Another thing, antibiotic goes in small amount, very good, it kills the bacterium, but my liver, usually all these chemicals go to liver and the liver breaks it down, and then it is sent out through either through the urine or through the motion as waste product. But when that molecule is broken down, suppose it's a molecule like that, and it is effective, it kills all the bacteria, it goes to the liver, it breaks into two. One of these portions is damaging the kidney. Then what is the point? So these are all the things one has to test. And in many cases, we find that the new so-called antibiotics is useless because of one of these points. And another possibility is you do so much of research and finally you get a new antibiotic, you see the molecular structure, it is the same as the one that is already in use. Replication, it's a waste. That's why people don't want to do work on this antibiotic research. So what we say is, okay, so here is one example. And then we also showed that this is in collaboration with Dr. Dinkar Sahal, this malarial bug. He's a malaria specialist in International Center for Genetic Engineering and Biotechnology in New Delhi. This we published in Parasitologica. Then we have another, we got some fungi from solar saltans. You know what solar saltans? Where the seawater is evaporated to get salt, which is sold, okay? Common salt, which is the concentration of salt is very high there. As you know, there are some fungi surviving in that environment. We call them hypersaline fungi. And some of them produce, we found that they produce some chemicals which would inhibit a very common human pathogen called Candida, which is there. You just show you see your tongue in the mirror, you see a white coat. Whatever you do with your breath, that white coat doesn't go. That's all Candida species. That's there. It's a common thing in humans. It doesn't affect you. It's there. But whenever immunity goes down, this thing will grow. It will multiply. It grows down the throat. It grows into the lungs. It fills the lungs, and lungs can't breathe. Die. So now they used to give antibiotic to kill this fungus. It's actually a fungus. It's yeast. But there are only, throughout the world, there are only 4 antibiotics. And now there are some of these Candida species which are resistant to all the 4 antibiotics. So you can't do anything. Recently, you must have seen this news of black fungus infecting people with AIDS from the respiratory thing and all that. That's all, they're all resistant. It's a very easy thing. It was an easy thing to cure fungal diseases. But now it is not easy because there are only 4 antibiotics and many fungi have become resistant to all these 4 antibiotics. So this we are trying to publish this paper. It is in collaboration with Dr. Kausu Sanyal and his student, Dr. Panchal, who are in this international center, this Jawaharlal Nehru Center for Advanced Scientific Research in Bangalore. Okay, so now I'm coming to the last maybe 3 slides. So what do we, based on our work here, what we suggest is what I suggest is you take drug-resistant bacteria, and then you take microorganisms from extreme habitats like desert soil, or I told you, endophytes from plants, or plants which are growing in high salt farm like mangroves. So take those fungi. Why we cannot take common fungi? Most of the antibiotics today, with natural antibiotics which we are using today, have come from soil. So avoid soil. Bring these fungi which are not been studied, or bacteria which are not been studied. Keep them in a place. Put these drug-resistant bacteria isolated from a patient. Put them there. There is going to be a fight. You create an artificial environment where MDR is allowed to fight with fungi from less explored habitat, and then look for novel antibiotics. We don't know. Here is one sample. I'm just giving you from our paper, the paper that we published. Take, for example, when you allow this reaction to take place, what happens is, so this is the drug-resistant bacterium, this black dot. So let us say, when we do this, you take fungus D. This is a fungus D, and because it is the harsh condition, high salt, high pH, or very low pH, or whatever, because of that, it produces a metabolite, an antibiotic, and then that can kill the drug-resistant bacteria. That is one method you can expect. The other method that we can expect in this reaction is, okay, so here is number 2. That is, there are two bacteria, A and two fungi, let us say A and C. They interact with each other, and because of this interaction, one of them produces an antibiotic which can kill this multi-drug bacteria. If only C was there, no antibiotic would be produced by that, and there is no killing of this. If only A were there, no antibiotic. If A and C are there, either because of the interaction between A and C, either C will produce a chemical or A will produce a chemical, which will be an antibiotic, and which you can expect it to kill this. Another possibility, there is a bacterium or a fungus called B, okay, and it interacts with A, and that produces a chemical. That chemical is not an antibiotic, but there is another bacterium or fungus D here, which will convert that chemical. It is called biotransformation. It just adds a group, a methyl group or a COOH acid group or something like this to this chemical. That becomes an antibiotic. So these are the interactions that one can expect. And that way, there are more chances of getting novel antibiotics than just by going and taking a soil and looking at it. Here we are creating an artificial warfare between the multi-drug resistant bacteria and microorganisms which have not been studied in detail. Bring them together, try. Of course, not all labs can do this, because you heard of safety levels in laboratories. The safety level 3 or safety level 4 can only use this because they are human pathogens. But this is an idea. Okay. What now? The other thing. The question is, why am I talking about fungi? Why we know all these examples, fungi. I am not talking about bacteria. The reason is, as I told you, many of the antibiotics which are currently in use are from soil bacteria, soil-dwelling actinomyces. Therefore, other types of microorganisms should be explored for novel antibiotics. And why fungi? There are supposed to be around 3 million fungi species existing. They have estimated that, but out of that, we know only 8%. So there are 92% of fungi remain unexplored. We do not even know where they are. And then fungal symbionts of marine organisms, deep sea fungi, those that are associated with mangroves, and fungi from extreme habitats like in Thar desert or someplace, or high or low temperatures. acidic soil, alkaline soil, and then salt stressed soils. You can try them. They produce different types of metabolites. Okay. Then, okay, you can ask me, right? All these things sound fine, but who is going to do this work? Because we already mentioned that pharmaceutical companies are not going to do this. Srinath, I need just another 5 minutes. Is that okay?
Please, sir, please.
Okay, excellent. So how do we do this? Why the pharmaceutical companies are not interested? They have to spend a lot of time in collecting these, high temperature, low temperature soil, acid soil, etc., and then isolate fungi. You get all sorts of fungi there. Then test each fungus whether they produce any antibiotic, and then take that antibiotic and see if it is good, if it is already not described, if it is a new thing. So they are not interested. So how do we go about doing this? So we suggested a model. You can look at this paper in Current Science. Venkat Gopal, my collaborator in my Fulbright host, we suggested a model, a crowdsourcing model. Yeah. So what we do here is, you take, let's take a college or a university which is located in a place like, say, in Andaman Islands, where there are a lot of mangroves, or in Thar Desert, where there are desert soil. So we can bring the faculty of those colleges or university to a lab and train them to isolate fungi and to look for antibacterial metabolites. We are not telling them to use human pathogenic bacteria because it is college, they cannot use, but they can use general non-disease causing bacteria. That is allowed. So we only, instead of my going to Thar Desert, going to Andaman mangrove, going to Kerala or Lakshadweep to collect coral, then going to Western Ghats to collect fungi from forest or from the dung of deer, spotted deer or something. I call these teachers from colleges located in these places, train them how to look for fungi, how to screen them for such antibacterial substances, and then send them back. What they do is they train the students. Most of you would be doing a project, right? In your BSc and MSc course, undergraduate and postgraduate course, you do a project. In that project, you can use this as one of the methods. And a register can be maintained by the college saying that this teacher asked in this project, 5 students joined together and they looked at the dung of let's say some herbivorous animal like a spotted deer or a bear or something like that for novel microorganisms and fungi, because we teach them how to isolate fungi from these substances. And then, so a register is maintained which year, and then the cultures are easily collected. We teach them how to collect and how to store it. After 5 batches of students doing this, that college, let us say, which is located in Andaman Islands, would have at least 500 cultures of fungi isolated from sea, marine source, sea water, sea fish, or water. And then the next 5 years, the students can be asked to screen them for, look for antibacterial or antifungal substances, again using non-disease causing. Then if something shows very interesting activity, that strain of the fungus is given to a national laboratory with which you would have already signed a MOU. Look, when we get this, you please take it up. So they can advance investigation. They select by industry or national labs. they select and they do the work. And so you have solved the problem. You have looked at hundreds of fungi and you have arrived at one fungus and given it to this lab. So you have reduced their work. So they'll be willing to see if it is a novel antibiotic. And then if it is a novel antibiotic, then they inform you. Then the culture of that is deposited in our national culture collection. And then finally, somebody would do a database search. And then if it's a really a new and effective antibiotic, the outcome, whatever, is shared by all the college teacher, the student who did that work, because we are maintaining a register. And then the industry or the national lab which did that work. So the patent money or whatever is shared by all. And also because you deposit that culture in a national fungal or microorganism deposit culture collection, you are adding to the genetic resource of the country. So it's a crowdsourcing model which one can follow to effectively isolate novel antibiotics from less microorganisms of less studied environment. So thank you all for listening, and my sincere thanks to Dr. Srinath for inviting me to give this lecture, and to Swami Sukadevananda Ji Maharaj, who was the Swamiji and Secretary of Ramakrishna Mission Vidyapith, and also the chairman of our Windstorm Lab, for his encouragement and for all the facilities that he has provided for us to do our work. So that's it. And I hope you liked the lecture, and I'm willing to take up some questions if you have.
It's fantastic, sir. Really nice. I mean, the last time I sat on your lecture was in Manipur, sir, if you remember. I mean,
yes, I remember. Yeah.
Yeah, that was the last lecture. That was really, really, really nice. Fantastic, fantastic lecture. So thank you so much. And I think I'll come back to you. I think there are a lot of wow, a lot of questions. But well, let's go one after the other. Okay. So Soham, do you want to go first? So people, just another reminder. Introduce yourself. Say what you're doing, where you're from, what are you trying to specialize, because that gives a perspective of the background of the question that you're trying to ask the professor. Suryanarayanan. Please, Soham, please, real quick.
Yeah. Namaste, sir. It was a mind-blowing lecture. I really loved it. My name is Soham. I'm from Maharashtra. I'm going to attend an offline training session about the microbiology in Kochi. My question is there any way to diagnose how susceptible we are to this drug resistance, how susceptible we have became from taking the antibiotics in past, or whatever the evolution might have been? Is there any way to diagnose it, any medical test or anything?
Yeah, we cannot have a general test, that will be too much. But supposing a person has a particular disease, let's say TB, then they take the sputum sample, the phlegm from the throat or from the coughing, the phlegm, and then they culture that and get the bacteria out, and then test for resistance. You then just have a petri dish in which you grow the bacterium and cut a well in the petri dish, put rifampicin, cut a well here, put isoniazid, and see where the growth gets inhibited. If it is inhibited by isoniazid, okay, isoniazid is good, and we can treat the patient with isoniazid. If there is no inhibition, it means for both, it means he's having a superbug or she's having a superbug infection. Then it could be a little more complicated. I don't know if there is any new drug for treating multi-drug resistant tuberculosis. Maybe there. I'm not sure. But we can test.
Yeah. For specific disease, you mean?
Yes. For specific disease, yes.
So I just have another question, if that's okay.
Please.
Yeah. Is there any mechanism that makes these organisms transfer between interspecies? Like, can a fungi give information to bacteria or anything like that? Excellent. And why do they do it? Like, let's compare it with humans. If we get under any stress, we would never share that information to someone who could benefit out of the same stressor. and these organisms, like I feel that they are altruistic in nature.
Yeah, this is what is known as intergeneric gene transfer. We call that horizontal gene transfer. So when the genes are transferred from parents to children, it is vertical gene transfer because of fusion of gametes. So it is, but not only in humans, in plants, it's vertical. You get a mango pollen grain falling on the stigma of a mango flower, and you get a mango fruit after fertilization, and it has a seed. That seed will have the genes of both the mango plant. It's not going to have a gene of a coconut plant. But now we are able to see that due to this bacteria, the bacteria can get not only bacterial genes from other bacteria, it can also take in human genes. It can also donate its gene to human body. If you take our human genes, human gene mapping has already been done, as you know. There are many viral genes there attached to our gene. So over the period of several millions of years of evolution, horizontal gene transfer has taken place. Horizontal gene transfer means gene transferred from one organism to another organism, which are totally taxonomically unrelated. Bacteria can transfer its gene to plant. Bacteria can transfer its gene to humans. So that can happen. Yeah, that has happened. You can get those genes in our system. You can scan it. I mean, if you look at the human genome sequence, you will see many viral genes there. There are many bacterial genes. They become a part of us. They are silent, but they don't express themselves. But they can be. You know, I read some recently that this COVID, it's an RNA virus, as you know. When they looked at the COVID virus gene, they found that some of these genes which are related to COVID-like virus, RNA virus, have become a part of human genes, and that those genes are responsible for forming the placenta between the mother and the embryo when the embryo develops.
Wow. Okay.
Yeah, if these things didn't happen, human beings mothers cannot bear children. Isn't that amazing?
And amazing and a little scary also, sir. In one
exactly. So some of these things are useful, but we don't know many of these, especially the internal microbiome. Please go ahead. And when you have time, look at the endobiome of our intestine, the bacteria that we carry. They have genes. Why a person, I don't know, are they taking it too far? I'm not sure. But there are indications. Why is somebody more reactive? Certain persons are short-tempered, certain persons are not. And like that, there are many things. The difference is they see it's because of the type of bacteria that they are having in the intestine, in their intestine. Many people get irritable bowel syndrome because of too much of use of antibiotic. One of the methods to treat that is, although it may sound very unpleasant, is to take the stool motion of a person who has normal bacterial population, take that, grow, and then get those bacteria, put them in a capsule, swallow like probiotics. All these probiotics are all such stuff. Of course, they're not taking it from motion, but they're culturing it. And then, so yeah, the horizontal gene transfer is pretty common.
Yeah, and again, sir, your solution, that's of course it's quite interesting, but it has its own challenges, sir, doesn't it? Because it's still a foreign.
Yeah, you're right. Unless some small initiative could, prove it to a little extent, I don't think they would be interested in doing this. But a time will come when they may have to think of this.
Fantastic.
Because we need a lot of people working on it, but nobody is willing to spend money.
Yeah.
This way the students gain their degree, they gain some experience, and if they're lucky, they also can be a part of a major breakthrough.
Yep. Or get into an IT job and then settle really well.
Exactly. Yeah. Yeah. True.
It's so strange. I mean, 55% of the previous batch students have all made application to IT jobs. And I'm just thinking.
Yeah, right.
But anyway, yeah.
Time should come, and I don't know. Here's the thing. To deal with antibacterial resistance, they go to IT. I mean, there's no other go. So they look for. I think now they're using huge data which they have collected, because bacterial genes are easy to what do you call it? To sequence, because they're small easily. So they do that, and then they collect a lot of data, and there is a lot of data bank, and they go and look for novel methods to control and things like that.
Absolutely, yeah. Absolutely, yeah. So if it's okay, we'll just take one last question, and I think Ritham, do you want to go? Ritham, please.
Yes. Hi, good evening, sir. Suryanarayanan, sir. Hi, I am Ritham Datta. I am from West Bengal. And
From?
West Bengal.
West Bengal, okay. Yeah.
And PhD scholar under University of Madras. Yeah. Sir, can you elaborate this method? Why you choose this particular seaweed? Because there are a bunch of seaweeds around this. Why you choose this particular seaweed?
No. You're right. We didn't choose this seaweeds. We actually, if you look at the paper, we screened more than 120 species of fungi isolated from brown seaweed, red seaweed, and green seaweeds, and then did a basic test. Out of that, some of them were effective. And then the ICGB chose the better one, and out of that 2 fungi, this Fusarium, and there was one more, which was the most effective. So we go step by step. You're right. We have to screen many.
That's right, sir. Thank you so much, sir.
You're welcome.
Absolutely fantastic, sir. I know there are a lot more questions, and I think if you want to write to Professor Suryanarayanan, you could because it's already.
You can email me. Yeah.
Absolutely, sir. That would be absolutely great. And sir, thank you so much. And I know my colleagues wanted to do a formal vote of thanks, which I would just leave let them do. But I just wanted to thank you really personally also, sir. I mean, this is a fantastic lecture, and thanks for a great opportunity. Because last time I sat there, it was absolutely amazing, because I just got into the lecture room when Professor Raman and you were actually giving the presentation. It was so nice, because I sat the entire 2 hours, and your last 2 slides where you just introduced the new ways of presenting graphs, interpreting data, they were fantastic. And I'm going to just talk to my friends about that as well, and then give a different perspective. But I think we'll go with the vote of thanks first, if it's okay with you, please. Yeah. Oh, yeah. So I think Srishti, the floor is yours, Srishti, please. Yeah.
Thank you, Shri. Good evening, everyone, and good morning to you, Shri. Thank you, Professor Suryanarayanan, for taking the time to talk to us today. I think we can all agree when I say that today's session was incredible. Antibiotic resistance is and your lecture has helped us understand the importance of these drugs and its usage. And I think it's very imperative that we take these drugs seriously if we wish to eliminate resistance in the coming future. As we know, knowledge can only grow if shared. For this reason, I want to wholeheartedly thank you, sir, for sharing this essential piece of knowledge with us. I would also like to take this opportunity to thank Shri and Mukesh. I know they both don't let this out very often, but truly they both do everything in their ability to help us out. Mukesh with not only modeling, but also, bettering ourselves. And Shri, well, without him, we wouldn't even have the opportunity to listen to this amazing webinar. So Shri, thank you. My last thank you I would like to give is to my YAP classmates. Thank you to each and every one of you for attending this webinar. I hope you can take what you've learned today and create a new and a fresh perspective. Finally, Professor, please give me the honor to once again thank you. We hope to interact with you again in the future, hopefully offline, with more inspiring contents. Thank you.
You're welcome to, please feel free to send questions to me by email, and if possible, if I can know the answer, I can inform you and maybe indicate where you could get some of these and how one could go about doing basic work in this field and things like that. So on the whole, I enjoyed because there were a lot of feedback and a lot of questions asked. Yeah, so I'm happy. Srinath, once again, thanks a lot for getting this done and bringing your people together. Thanks a lot. Good luck to all the students.